today it's my absolute pleasure to announce that our faculty composes of Jeff Barrett from the Bill and Melinda Gates Medical Research Institute. Jeff is the head of quantitative sciences at the Gates Institute, Gates MRI Institute, and we also have Kyla Andrews. Kyla Andrews is a scientist in the Translational Discovery Group, and today they are going to take us through the pharmacometric considerations for guiding pediatric trial designs. And I want to thank them for their time and in agreeing to do this webinar. So I just want to say again that we will, they'll take us through this webinar, and at the end we will have a chance to ask questions the last 15 minutes of the webinar. So please keep those questions. Also, the webinar is going to be recorded, and the recording will be made available by the end of the week. So in case you have colleagues who have missed out and would like to review it again, we will make the recording of the webinar available. But thank you very much. And now I would like to hand over to Kyla and Jeff. The floor is yours.
Thanks, Ada. And again, on behalf of Kayla, this is a real pleasure to be able to spend the next hour or so with you all and share some of our experiences supporting pediatric development programs through pharmacometric considerations. And you know, a lot of what you'll hear today from us is related to actual clinical trials, some of which a little bit dated in terms of the actual use cases that you're going to see. Many of them are more than 10 years old, but I think it provides a nice historical context. And just as a way of reminding you all, this was at a very pivotal time in pediatric drug development where the FDA was issuing guidances around pediatric drug development. A lot of the actual approaches that you're going to see are also dated with respect to some of the software and tools. We're going to share all of the code with you, so you'll have access to all of the behind the scenes methodologies and approaches, and we're happy to dig into any of the details, but for the most part, we're going to present a high-level view of how these approaches and strategies line up to supporting these clinical programs. So we're going to start with a little bit of background in terms of what is a context for supporting clinical investigations, specifically in pediatrics. We'll talk about this from the standpoint of a pharmaceutical sponsor and highlight some of the codependencies and interrelationships. And we'll talk about the variety of approaches and methodologies that are available to you as the modeler before you engage in developing a strategy to support the team. And then Kayla will take over and walk you through the details and a lot of the behind-the-scenes work that led up to two case studies. So remember, you know, pediatric clinical investigation doesn't begin with respect to the necessity of doing trials prior to a pharmaceutical sponsor doing a lot of research, describing a molecule, developing a signature around the attributes of that molecule, pharmacokinetics, pharmacodynamics, the basic physiochemical properties. A lot of this is matter-of-factly generated as a sponsor engages in early-stage development. In many cases, in most cases, in fact, a complete adult program actually precedes investigation into the pediatric initial trials. So as a modeler, you inherit a lot of the data that is forwarded to you from the pharmaceutical sponsor so that you do have things to work with. That also gives you a starting point from which you can develop a strategy. So in this slide, we kind of highlight some of the issues in each of the programs that makes you really have to think very difficult about the data that you're going to inherit. So particularly not all data is of equal value, so you as a modeler has to spend some time looking at the quality of the data, the integrity data, the conditions on which it was generated, be that in preclinical, phase 1 or phase 2. All of this would roll up into what would be the starting point from which strategies develop. One of the other things that we usually inherit, though, is the lack of pediatric-specific knowledge on formulations and information about the therapeutic windows, so that many of these initial studies in children often reflect the need to fill some of the gaps in that knowledge and precede actual clinical investigation. You know, the other thing that we see is when we talk about those initial studies in children is we're often constrained by limited sample size, difficulties in actually enrolling patients. So that's some of the reality about doing pediatric trials in general. For the most part, the initial foray into pediatric investigation is building confidence around the therapeutic window within children. So understanding the safety, tolerability, basic information about the pharmacokinetics and pharmacodynamics, with the expectation of making comparisons to what has preceded in the development of the adult indication. But the primary emphasis is on safety. This is something that I think we always have to remind ourselves of, even though, you know, as a modeler and potentially clinical pharmacologist, you're desperate to fill in that gaps. From the sponsor and regulatory standpoint, it's really about demonstrating adequate numbers of patients' exposures. So this compromise between studying enough patients to get confidence and also enough patients to formally study the exposure profile in these target populations is a part of the issue here. When we move into phase 2, of course, we are uniquely focused on finding the dose in that target population. Again, I would say there's often a bridge between what has preceded in terms of experience in other pediatric development programs, but also in adults. And likewise, it would behoove the sponsor to develop pediatric-specific biomarkers to support the dose, but also be the basis for which efficacy can be demonstrated. So there's a building set of information and knowledge that evolves as we start to fill in some of these gaps, but also the models kind of facilitate this and cement our understanding around the attributes that we say the molecule does have and should have for it to be effective in children, and also information about what we need in terms of the proposing future trials. So a lot of the time, and it's highlighted in that middle box there, is we need to respect the fact that you know, these are children and not healthy volunteers. There's issues around how much sampling you can actually do from a regulatory standpoint. And we don't want to really oversample, both in terms of the amount of blood that we draw, as well as the number of patients that we study. So we need to propose a design that has the greatest information value and still allows us to fulfill our objectives in the protocol. So again, some of the obstacles along the way is that we don't always get the same kind of information depending on the molecule we're talking about. So the issue at the very beginning is really understanding the landscape of that prior knowledge and doing an inventory of what data I'm actually going to inherit as I start this exercise. And then being able to educate those around me in the project team and potentially the institutional review board who would be able to look at this output and judge if it's fulfilling its purpose in terms of enough information to be useful, but not over information in terms of the number of subjects and the extensive sampling. So being able to operate with these practical constraints, but still deliver good information in terms of providing guidance around the gaps in our pediatric clinical pharmacology knowledge, as well as delivering eventually dosing rules that allow us to recommend these medications with confidence. But the methodology, I want to make this point, methodology is really not the issue. We have a lot of approaches here. There's good historical precedence. I would strongly recommend that those who are engaging in this area read the literature and get comfortable understanding good case studies. Again, as a context for where we begin, the clinical exercise really starts at the level of the protocol. So there's a lot of information that is expected from the sponsor to be filled in here. And I think the key information from the standpoint of the modeler that we try to touch is informing the study design, providing a good rationale for the dose in children with some relevance to the adult experience, the details around the number of dose groups, duration, all of this kind of rolls up into a signature that allows us to say with confidence that we understand the therapeutic window, and the modeling piece of this is really just a tool. What we're going to measure, that's also linked to how often we measure it and the volume of blood that we draw, as well as the age strata. So some of this is also defined by the intention on the indication. Certainly the sponsor will have thought about, the age brackets in which they perceive this to be useful, and that will guide us in our exercise in terms of the simulations that we take forward. And of course, this planning involves a lot of other a lot of other disciplines that come up and assist us in this exercise. And also there's a lot of dependencies around these other groups. So things like the adult effort around the pediatric, around the development program, formulation considerations. Again, we would like to have some information around the population of interest, so we'll pull in thought leaders who are representing that therapeutic area. So if it's a drug for diabetes or some cardiovascular indication, this will not proceed without getting input from people that are typically actually dosing those children. So we know something about the disease progression. We understand the constraints for how these populations exist and how they're cared for. We also know something about the prevalence rate. So, and the timing on when this information is available is also critical because, again, sometimes we have to proceed without the information we'd like to have. So we have to operate with knowing that there are some gaps. In terms of the dose selection, we would love to be in a situation where we had a very simple dosing rule about how we achieve making that initial prediction of the dosing requirements in children across the age strata. But we often find, and it's well appreciated, that a lot of the simplistic rules just don't perform well across all age strata. So a simple rule just doesn't exist. So things like just simple linear adjustment by scaling to the adult body weight underpredicts across all age strata. Using body surface area is very commonly applied in oncology, but it still underpredicts in both infants and neonates. And even something like an allometric rule, which takes care of some of the nonlinearity of the first two rules, does have a tendency to overpredict children less than 1 year of age when it's used by itself. So none of them are perfect, but they all give us some indication of a general dose requirement. But it is really incumbent upon the modeler to eventually deliver guidance when we actually conduct the study. So this is always a situation where we're modeling and simulation works in tandem with the actual collection of data. And again, just as a reminder here, I said this previously, but we don't want to exsanguinate the child when we're doing these trials. So a key part of the deliverable is, again, to recommend a sample collection strategy that allows us to compromise between the information value we know we have to re-estimate the pharmacokinetic and pharmacodynamic parameters, but also to not oversample to the point where we're butting up against the regulatory requirements on blood volume. So this is a table that just gives you an indication of what the typical blood volume requirements are, and this is directly from the Children's Hospital of Philadelphia. That supported a lot of my work in the past. So this is what was negotiated with the Institutional Review Board across the age continuum. So you have to keep this in mind, and it also is obviously something that impacts those developing the bioassays. And as I alluded to, we don't always inherit all of the information we'd like to have. So what we do inherit kind of also gives like an indication for what type of approach and methodology we will employ. The two case studies you're going to see are going to be from a classic top-down approach that's in the bottom part of these concentric circles, where we actually get data in the population of interest and do a related pediatric indication and adults. In some cases, and it's particularly true in more recent examples, though, we will also inherit the physiochemical properties around the active drug substance and potentially the metabolites, additional preclinical data around animal pharmacology, other information that tells us information about the therapeutic window, such as the toxicology data. So all of these would be considered priors that could be useful in an informing approach. Of course, depending on what you inherit, that obligates you into moving into certain certain methodologies and approaches versus other. But we're going to show you examples that are, again, going to be in the top-down approach. This is going to obligate us to using and leveraging the prior knowledge from either previous pediatric studies or adult studies. And typically, we'll use a classic pharmacometric approach using tools such as non-mem to develop the initial structural model and adjust for the nuances that allow us to predict behavior in children. One of the other factors that often comes into play as we're considering what approach to recommend to the project team is this relationship between the fit-for-purpose approach and actual pragmatism. So again, as I mentioned, we have to deal with these constraints in terms of the available population of interest. It's not like we have a phase 1 unit that studies children in this regard. So we're typically talking about children that have a very specific indication. We will have to go to some typical academic medical center where these patients can be found and where there's an investigator who's involved in treating them. And also marry that up against the blood volume constraints, so I can't oversample this population. So we have to also pick approaches that allow us to establish with confidence a methodology that allows us to deliver on these, our intended efforts to support the protocols. The typical procedure in this top-down approach will require that we have some knowledge of the adult information. So we do have an adult reference point to start with. And we'll use that as an anchor that allows us to know what is the appropriate signature, be that pharmacokinetics or pharmacodynamics in adult. What is the target that we're trying to achieve in children, particularly if we're taking an equivalence type approach. And we will use methodologies that allow us to modify the adult signature and make predictions in children. So there's a framework that starts on how we characterize the adult information, and then an adjustment to this so that we can actually use a simulation approach to actually project what's happening in children. And again, we'll use this iteratively to drive the generation of actual pharmacokinetic or pharmacodynamic metrics. We'll make comparisons across different age strata, and we'll be able to look at how this rolls up into similar or not PK and or PD responses. So we're generating an adult signature, we're modifying it to predict in children, we're predicting pediatric behavior across age strata, and we're also taking into consideration when we're taking data. So the case studies will show you how this works in this iterative way, but this is kind of a general description of how all these methodologies roll up to do this. And again, we're going to make adjustments to some of the structural components of these models to adjust for additional factors such as maturation and organ function and other patient-specific factors. I won't go into detail about a lot of this adjustment, but there's a huge body of literature to guide you in this regard, and happy to answer questions. So with that, I will turn it over to Kayla, who will walk you through 2 case studies illustrating this approach.
All right, thank you so much, Jeff. So we're going to be talking about 2 different case studies today, both of which we've used modeling and simulation to support study designs for clinical trials. The first one is for DALTEPARIN in pediatric patients, where we specifically were looking to help inform sampling scheme and sample size for each of the cohorts in the proposed clinical trial. And the second case study focused on FLURBIPROFEN in pediatric patients, specifically the formulation and sample size for each of the cohorts. So for our first case study, this involves DALTEPARIN. DALTEPARIN is a low molecular weight heparin. Its antithrombotic activities, the preferential potentiation of the inhibition of coagulation factor 10A. For the most part, this is a therapeutic agent that's primarily used in adults to prevent deep vein thrombosis, or DVTs, typically after surgeries like hip replacement surgery or such, when patients have severely restricted mobility. So this drug was initially approved in 1994 for adult patients, but actually very recently, in May of 2019, after we started preparing for this webinar, actually, there was approval of DALTEPARIN in pediatric patients for use in children 1 month of age and older. And you can see here, I have a screenshot of the label, and we have the indication noted and highlighted in yellow. So this is really exciting because this is the first and only anticoagulant indicated for pediatric patients. So this is very exciting. Now, I'm just going to take you a little bit back in time. So between the time that this drug was initially approved by the US FDA for adults, all the way up until 2019, there was undoubtedly much work done between this time. And the analysis that we're presenting today happened somewhere on this time continuum between these two dates. So what we're sharing is just part of the story before DALTEPARIN was approved for children. The sponsor was seeking an indication for this drug in children with cancer and wanted to design a dose-finding phase 2 study in kids to show non-equivalence to the existing standard of care. So we had a historical data set of this drug given in children without cancer. This was available for use. And the goal of this analysis was to support dosing based on those exposures from the historical data set. So, you know, we assumed the drug was safe, though we weren't necessarily sure if this was efficacious. Now, an important objective of the proposed trial is the investigation of PK/PD behaviors of DALTEPARIN in children, with the goal of characterizing dose and exposure response relationships. So the ability to define these quantitatively really depends on key design elements of the timing and the frequency of sampling, as well as the number of samples obtained per patient. So these can be, you know, very difficult practically in obtaining samples in children, given the volume required, the timing of these samples, given that these children have other procedures that are going on. And as Jeff mentioned, enrollment can be very difficult as well. So because of this, modeling and simulation was used to recommend sample size and sampling schemes for the clinical trial a priori so that we could really try and get the most that we could out of the data for this proposed clinical trial. So in order to achieve this, we had a few objectives. So first, to use the previously developed PK/PD model to simulate various sampling schemes and sample sizes, to evaluate summary statistics on the precision and bias of PK/PD parameters for each of the data sets that were simulated, and then of course recommend a sample size and sampling scheme for the clinical trial. So again, we're trying to use modeling and simulation to propose the minimal sampling scheme that will get us the information that we need. So in addition to the challenges that I briefly mentioned, you know, there are also some other concerns. So the sponsor had a preference to use a single point design. I'll tell you a little bit about that in a moment. But this preferred small sample size and sparse sampling really presents challenges when anticipating regulatory concerns. Making sure that we have enough subjects to assess the PK safety relationship. Do we have enough subjects within each age strata to test for differences across groups? And we really need to make sure that we have adequate sampling to determine the critical PK parameters within and across age strata. So with all of this in mind, we asked the question, can we propose a sampling scheme and a sample size that takes the least number of samples and lets us make the most efficient use of the data? So the basis for the simulations was the POP PK/PD model developed from an open-label dose-finding trial in children that were older than 36 weeks of gestational age up to 16 years. There were 31 children total in the data set. And the measurement of 10A activity in plasma served as a surrogate for the concentrations of molecules that contain the high affinity binding site for antithrombin 3, so the anti-10A activity. And this is because there's no mass spec assay that would enable us to look across all of the low molecular weight heparins and be able to have these plasma concentrations. So we used this as the anti-10A activity as a surrogate for activity, okay? So here just have some demographics of the historical pediatric data. So you can see that children were included as young as, you know, just over a month up to a little over 16 years of age, and they weighed on average 22.6 kilos. Now, the really nice thing about this, this historical data set is we had really rich data. So you can see here a sampling density from this data set. So the x-axis shows different age bands, and the y-axis shows the number of samples. So the key basic information here is that you have a lot of data across the different windows from neonates to adolescents. And this is really not characteristic of pediatric trials. So this is a unique data set, and this was key for us to be able to develop an initial POP-PK model off of the data. So here, just a quick overview of the POP-PK model, trying not to get too technical, just a little summary here. Two-compartment model best describes the data with first order absorption, clearance and volume were allometrically scaled, we had a proportional CV error model, and we had endogenous anti-10A activity, as we mentioned again, as a surrogate for the concentration of the low molecular weight heparin DALTEPARIN. So in the estimation, NOMM was used with FOCE with interaction, and here you can just take a look at the final parameter estimates and their relative standard errors. You know, overall, the POP-PK models, parameters were pretty well estimated, considering there were only 31 patients, and the relative standard errors are rather small. And this is really a byproduct of having a very good sampling scheme across the whole age strata. So we were quite happy with this POP-PK model moving forward. Just to give you an idea of what some of the goodness of fit plots looked like. Here you can just take a look at the diagnostic plots. So you can see, for example, our population predictions versus our observed data. There's a little bit of a bias here. So we have a little bit of under prediction. But then if you look at the individual predictions and the observed data, you can see very nice concordance across the line of identity. So we're quite pleased with this. I have some other yellow circles here on the screen, and I just wanted to highlight to you that these were all of the same two patients. So overall, when you're looking across the weighted residuals over time and the weighted residuals over the predicted values, you know, these are pretty good plots. So we were very happy and confident moving forward. So with the approach that was used, again, we're trying to evaluate the effect of the sample scheme and the sample size per age strata, which we have here shown on the screen, on the basis of these key PK/PD parameters. And we're really just trying to establish that dose exposure response relationship between children without cancer, which was our historical data set, and with cancer, which is where we hope to conduct this clinical trial in. Okay, so in addition to looking across the age strata, we were also looking, of course, at the sample size. So we had different numbers of patients in each age strata, and therefore also a different total number of patients that we were evaluating. And for each of these different sample sizes, we were also evaluating single-point sample designs in addition to two-point sample designs. And here you can see the list of, for single point, 4, 7, 12, and 24 hours, and for 2 points, 2 and 6, 4 and 10, 3 and 8, 5 and 12 hours, and of course this is all post-dose. So you may be wondering, you know, why such sparse sampling? Well, you know, again, we have to remember the patient population that we're working with, and a lot of this is a real case study, so a lot of this is dictated by the belief by the sponsor that, you know what, it's going to be very difficult to enroll these patients, and we don't want to be poking and prodding them all the time. We want to see what is the least number of samples that we can take in order to get the information that we need. So in order to complete these simulations, you know, there were a series of sequential steps that were conducted. So first, of course, creating the data set based on the desired sampling scheme and protocol dosing and design, which for your reference is on the previous slide. And then writing simulation code for each of the data sets using the previously developed POP-PK PD model, and then for each of those simulation data sets, we were looking to run 100 replicates. And so those simulations were run, and we had 100 data sets for each of the scenarios. Those data sets were fit using NOM-MEM and Perl to generate the model fits from each of the data sets and for post-processing. And then, of course, looking at the bias and precision of these parameters that are coming from these data sets. So a macro in SAS was run to evaluate each of these PK/PD parameters, and then of course we summarized the results to be able to look across, stratifying by sample size and sample scheme. And in case anybody's wondering what S+ is, that's sort of how you would think of R today, okay?
Commercial version.
Yes, it's just the commercial version. So here we are with the results. First, looking at sampling density. So the single point designs, those were really not successful, unsurprisingly, right? They didn't have acceptable bias. In most cases, NOM-MEM couldn't fit. It just had trouble converging. So none of that is presented here. What we are showing you are the 2-point designs, 3 and 8 and 5 and 12. Of course, you can see from the graphs were performed a little bit more efficiently. So you might be wondering why the 3 and 8 and the 5 and 12 and the 2 and 6 and the 4 and 10 are paired together. So here, the patients were ultimately randomized to get either or, okay? So for the 3 and 8 and the 5 and 12, we had patients that were randomized to get either the 3 and 8 sampling scheme or the 5 and 12 design. That was the ultimate goal of the clinical trial, so that's how we're presenting the data here. And so if you take a look at these graphs, on the x-axis, you have your PK parameters, clearance volume, and rate of absorption. On the y-axis, we're looking at percent bias. And we're really looking around, we're hoping for a 0, okay? And we have highlighted maybe about 50% above or below that as that light yellow box. And you can see that the 3 and 8 and the 5 and 12 are really falling within there much better. So for the sample size results, here, we're seeing the sample sizes of N of 20, 30, 40, and 50. N of 10 was not shown because, it followed the trend. The results were a little bit worse. There was much more bias and much more precision. Overall, if you're looking at these, the takeaway is in order to improve the precision of these PK parameter estimates, you really need a bigger sample size. So here what we're looking at, again, clearance volume, rate of absorption, on the x-axis, percent bias, on the y-axis, we're seeing that for N equals 50, which is 10 patients per age strata, we're really hitting the bias that we're looking for. So as you can see from the figure, the interquartile range for each of the key parameters really evenly brackets the 0% bias designation, and it's within about 20% for clearance, which is great. So just to wrap up this case study, the results confirmed the appropriateness of the proposed sampling density and sample size for the clinical trial. These were derived from the simulations and from the modeling, and they were ultimately integrated into the protocol sections for the clinical trial. And so, this is a clear example of when modeling and simulation was able to inform a clinical trial design, which is very exciting. In terms of after the execution of the clinical trial, what was agreed upon was a series of steps, which included POP-PK analysis based on the completed trial. This would inform the dosing guidance in pediatrics, fulfill expectations from the regulatory agency regarding the submission, and a series of other exercises which would really strengthen the recommendation for the dosing guidance in pediatrics. So moving on to the second case study, we're going to switch gears a little bit now, and we're going to consider pediatric formulation development for an NSAID. So this is the usual scenario. You have a lot of rich adult data. The sponsor is interested in developing the drug for children. For this case, the sponsor was interested in feasibility of an oral formulation for kids. So for those of you who have taken NSAIDs before, or if you have children that have taken NSAIDs before, they may taste a little bitter, so kids can have trouble swallowing pills. So the idea here was, let's get a formulation that would be more palatable to young kids. And keeping in mind that the bioavailability of a drug with which a child doesn't take is 0. So the formulation was really the key of the heart of this analysis. So FLURBIPROFEN is an NSAID that inhibits COX-1 and COX-2. It provides pain relief. It's primarily used for the relief of pain for rheumatoid arthritis and osteoarthritis. Doses in adults are given in divided doses. This was first approved in 1988. And, you know, despite the relatively broad therapeutic window in adults, there are no approved pediatric indications and dosage forms in children. And that's mostly because, you know, part of the challenge is, again, it's difficult to study pain in children. You know, this is mostly subjective. There's no biomarker to bridge adult and pediatric pharmacodynamics or outcomes. And, you know, in light of this, the sponsor chose to do exploratory analyses, assuming that these challenges could be resolved. So this project was reliant on picking a pediatric formulation which best mimics adult PK/PD expectations. The sponsor was not looking for a different indication. It was just, they were on the quick path to approval using an equivalence approach. The sponsor sponsored phase 1 PK safety trial with the goal of demonstrating equivalence to adult exposures. And there was historical data in adults which could be leveraged. So the goal here was to support study design for the phase 1 PK safety trial, focusing on equivalence and an equivalence strategy to adult exposures. In order to achieve this goal, there were some objectives here. First, developing an adult POP-PK model, scaling that POP-PK model, adjusting for expected age size differences across the FDA age strata, simulating various doses, formulations, sample sizes for each of those strata, evaluating non-compartmental metrics relative to the adult values, and recommending feasibility for sample size and sampling schemes for the clinical trial. So again, this particular case study, this was more feasibility work than anything. So here we have the methods and workflow outlined again. You know, very typical. We're developing the adult POP-PK model. This counts for different demographic and formulation effects. We're scaling this in accordance to these different expected age and size differences across these age strata. Again, we're writing our simulation code to create our virtual simulation data sets. We're running those simulations at multiple doses. We have 100 replicates for each of, or 500 replicates in this case. The last one was 100 for each of the dose groups and the study designs. And again, we're comparing overlap of these non-compartmental metrics, exposure, which is, you know, of course, the AC, the maximum concentration, Cmax, and the time to that maximum concentration, Tmax, relative to the adult values. Okay, so here, again, we're outlining the approach. And I want to focus your attention at the different age strata here. So this was pulled directly from the FDA guidance and was used for this simulation. The interesting part about this simulation is the formulation. So as you'll see in the following slides, which I'm going to flash through very quickly, and the size of the font is going to be small, but no worries. You will have the code and you'll be able to dig deep into it. Nonetheless, what's very exciting is the comparison of the formulation. That was pretty cool. So we looked at tablet versus lozenge. And so here you can have an idea of what was evaluated for one dose. So we have the different number of patients, the different formulation, and then the simulation scenarios were repeated for the 5, 6, 7, 8, and 8.75 milligram doses. Okay, so of course, first we developed that adult POP-PK model. Here's a screenshot of the NOMM code. Don't worry, you'll have a copy But what I just wanted to highlight here is, the indicator that was used for the formulation to examine the different fixed effects and relevant PK parameters in accordance to those formulations, how we're deriving the NCA PK parameters, et cetera. So then we had the creation of our virtual simulation data set. The adult PUPPY-K model was allometrically scaled to adjust for size. The priors of the adult model were used to drive the initial simulations. Of course, that this is the allometric scaling. We need to take this into account because if you don't adjust for this, then of course our exposures would be that we would see would be too high. So after the simulations were run, what we did was overlay the adult data on the simulations for each of the doses that were simulated. So let me just walk you through this figure. So what you're looking at on the x-axis is time in hours, and on the y-axis is plasma concentration in micrograms per mil of FLURBIPROFEN. So the solid black dots are the observed adult dose of 8.75 milligrams given as a lozenge. So it's the observed adult data. And then if you're looking at these lines, either solid or dotted, you can see a key at the bottom to show the different age strata. So these are the actual simulations that are coming from the analysis. So we're overlaying the results of the simulations on the observed data for the purposes of comparing, right? So the reason why the 8.75 milligram dose was chosen here is this was a dose that the sponsor said, we think we need to match the effect seen in the 8.75 milligram dose that's given to adults. We think that that's what we want to target. Those exposures are the ones that we want to target in kids. So what dose will match that? And so here you can see that these exposures in neonates and children are much higher than those that we're seeing in adults. And of course, as the age increases, you get down to the 13 to 18-year-old band, we see a more similar profile here. But this is giving us a first indication that, things aren't really lining up as we'd hope them to. So here, what we're looking at, we're going on to the NCA parameters. What we're looking at here is Cmax. And of course, so Cmax we're typically looking at for safety, right? In addition to the exposures, and we don't see as drastic as a change between the age groups as compared to the Cmax that we see in adults from the 8.75 dose. These are the simulations here of the different age strata, and this is all for the 3 milligram pediatric lozenge. So we're comparing the 3 milligram pediatric lozenge, the Cmax across these different age groups to the 8.75, and we're seeing that we're not getting that Cmax, but there's not really much of a difference between these age groups. Now, if we're looking at the Tmax, we can see that this is decreasing with age here, and once we get to that higher age group, it's very similar to the adults. And then when we get into the AUC, right, like this is what we're really after, these exposures. And here you can see that this AUC age-weight relationship is really difficult to dial in with a fixed dose across an age strata. And that's what the sponsor was after. They were after a formulation, fixed dose, that you could give across these age strata to achieve these desired exposures that they had confidence in from the adult data. And you can see that the exposures that were seen from the 8.75 milligram lozenge in adults, if you look across the different age groups in children, you're really missing the mark here. There's not a lot of confidence that these small children that are receiving the 3 milligram lozenge are going to get the same exposures as these older children. So this really provided a no-go decision to the clinical pharmacology managers to say, all right, this fixed dose in this formulation is not going to get us where we need to go. We got to put a pin in this, and we got to come up with a new strategy. All right, so in this case, you know, we see that the modeling and simulation gave us a no-go decision, and that's okay, because that's just as important as a go decision. And, you know, what we saw was that it was difficult to match the adult profile from a bioequivalence PK perspective without modifying the formulation for pediatric physiology considerations. And so this, of course, is going to be unlikely to convince regulators that there's a pediatric PD bridge. And I'll show you on the next slide, you know, that this wouldn't pass the FDA decision tree requirements, which predicates, you know, similar disease progression. So unfortunately, you know, this was a no-go decision, and there isn't an oral pediatric formulation for FLURBIPROFEN even today. However, there are alternatives for different formulations of NSAIDs for children that are weight-based dosing, not fixed-based dosing. So just wanted to flash this up here. I have the reference, which at the bottom, which you can you know, dig deep into later. But the takeaway here is that even if you pass the first question of, is it reasonable to assume that children compared to adults have a similar disease progression in response to the intervention, which is the assumption going into the analyses, then you conduct these analyses to be able to answer the question, you know, is it reasonable to assume exposure response in children compared to adults? And, you know, we saw here the answer is no. So in either case, really what this is telling us is that you would need PK/PD studies in these children to be able to understand that exposure, that dose-response relationship. So just to wrap up here, making sure we leave some time for questions, let's not throw the baby out with the bathwater, right? I mean, modeling and simulation has supported pediatric research and development, and it's made great strides. And since the time of this work, we've really moved pretty far in terms of the types of modeling that now can be used to inform dose selection, such as physiologically based PK modeling. You know, this has been incorporated into the 2014 draft FDA guidance. There are guidances on PBPK modeling themselves, and this type of consideration of transporters and expression of CYP enzymes and different maturation of organs and all of that, that mechanistic scaling mechanisms, this has really advanced our field. And so it's very exciting. So just to wrap up, you know, sometimes, even if you do use PBPK modeling or if you use allometric approaches, you get to a no-go decision, and that's okay because you just want to answer the question at hand. So with that, we are going to open it up for questions.
I just wanted to add that the no-go decision can somehow of money and millions. So now that you've opened the question session, Roland sent in a question. If you could move over to slide 30 or 31, about there. He was wondering on the drug that was used for in case study 1, was the precision and bias pre-specified, and what was deemed acceptable, and there were differences made, and were these differences made acceptable between parameters, for example, clearance versus Ka.
So great question. I think the main thing, that it was not pre-specified in terms of the team and in the protocol, nor did we get comments back from the FDA in that regard. We did prioritize the minimal bias and good precision around clearance and volume as opposed to Ka. I mean, I think the other thing to keep in mind with anti-TENE activity of low molecular heparins is there's an expected concentration in adults that is really the anchor for this, and it's at a 4-hour time point. So this is why oversampling to get better information on Ka was really not an objective and not anything that we think we could even do in the clinic.
Okay, thank you, Jeff. I'll go to another question. You've mentioned that you didn't get feedback from the FDA, you didn't get comments, but Roland sort of has a follow-up comment that does the FDA or EMA provide guidelines?
Guidelines on pediatric dose selection?
for this sort of thing, yes.
Yes, so we have that as one of the references in the presentation, and I can send you the hard copy of that can be circulated to all attendees as well.
Okay, thank you. Any more questions coming through the chat? I'm aware that I think Colin had a question.
Ida, so someone sent me a question privately. Jeff and Kayla, it was so good to hear your voices, and thank you so much for a wonderful presentation. So Kayla, this question is for you. I think it came from somebody that was part of the course that is currently running. It's scary that there are so many software to learn. SAS, S+, non-mem, R, Perl. Is that really all necessary?
That's a great question. So the answer is no, it's not all necessary. So in these examples, there were a lot of different types of software that were used simply because at the time, there wasn't an open source software such as R that was able to execute all these different types of modeling and simulation analyses. So I would say that your best bet for learning a software that enables you to execute these analyses would be R. So R has a lot of packages that you can do nonlinear mixed effects modeling, you can create all of these graphs, you can look at bias and precision, you can do exploratory data analyses, you can do simulations. So I would advise the individual who asked this question to focus on R. It's open source and it's always evolving. So that's what I would go with. Jeff, did you want to say anything?
Yeah, the other point I would make is, again, as Kayla's mentioned, the methodologies and approaches in addition to the software always evolves. So you have to keep in mind these solutions are not static from that standpoint. So even things like some of the scripting language, like Perl, for instance, was used very commonly in the day because of the need to automate and do things expeditiously. But now we have, with the improvements in high-performance computing, in addition to the software evolution, there's alternatives, as Kayla mentioned.
Thanks. Ida, I have one more, and this one is to Jeff. So Jeff, on slide 11, you presented the blood volumes. These are on the one hand small, but also large. How does this relate to actually analyzing the data? For example, if you needed to do a non-compartmental analysis. This question again came to me privately, and I think this is from a clinical person in the group that's attended the webinar.
Yeah, so it's a great question. It's very practical. I think what you have to keep in mind here is, particularly when you're going to go down to neonates, you see how the volume requirements shrink. So this really obligates the modeler to take a look at, well, if I have an existing assay, you know, what is the total number of samples I can take given the current volume constraints around that assay? And this may be a conversation that the modeler has to have with the assay developer. Can you improve the volume requirements? It's also a consideration that you may look clinically at, is there an opportunity to get scavenged samples? So things like heel sticks, other things that are going to happen coincidentally where you could kind of potentially use that sample for dual purposes. So I think, you know, you're always in a situation where you have to work backwards. You know, as the child gets older, some of the issues are less, but remember also that if we're talking about a separate venipuncture, the pain associated with that is very dramatic for the child. So we don't always assume that there's going to be an angiocath put in, and also to always think about minimizing the number of samples.
Thank you, Jeff. There's just another question. You haven't alluded to this explicitly in the examples you've used, but it's known that conditions such as malnutrition and then biology statuses like enzyme maturation of the liver play a role in drug metabolism, and therefore the concentrations that we see in children and neonates and across the different sort of age bands. So can you speak to how sort of if you usually make sure that you always include this in your simulations where you have different age groups? Can you just speak to that sort of physiology with maturation and statuses like malnutrition in children?
Yeah, no, it's a great question, and again, I would say it really depends upon what your target population is, and is that a prevalent factor that needs to be a priori considered in your model. So I pulled up the slide just to show you the question marks there identify these other patient-specific factors there. So there are adjustments made not just to clearance as well. So I think it's, again, a requirement of the modeler to think about this target population and adjust a priori. And there is probably also a stage that you have to consider in taking a look at, what is the range of expected values I'm going to get. This is always a situation where you can't predict things outside of physiologic commonality. So I think, yes. And the other thing, too, is that I'm showing you a basic top-down approach with classic pharmacometrics. It's potentially much easier to consider these kinds of real-world adjustments in a more mechanistic approach. But definitely it's relevant.
And if I may add something on there that sort of circles back to the previous question with all the different software out there, when we think about approaching this with a more bottom-up approach, there's software out there that there are models in which different compartments of organs and considerations of CYP enzyme expression are considered therein for children of different ages, such as SimCYP or GastroPlus, et cetera. And so there's very mechanistic modeling software out there that there's the opportunity to use and to explore. But again, it's all about the question that you're answering, and will the added layer of information help you answer it better. In some cases, if we're looking at a drug that's, let's say heavily metabolized by CYP2D6, you may want that information. You might be able to incorporate that in either approach, top down or bottom up. It totally depends. It's up to the modeler to make that decision.
So Kyla, we have one last question. It says, how did your model minimize the carryover of bias arising from gender and age-associated differences in plasma? And he goes on to say, would a semi-mechanistic approach be generally better? And you've sort of alluded to the fact that indeed, depending on the situation, you can use a more mechanistic approach to sort of account for the different biases in terms of age.
Right. So one of the things that could be done in a model is to put gender effects on different PK parameters, such as weight and a weight effect on clearance. In this case, that wasn't done, but it could be done. It all depends on how informative the data is. So, typically in PopPK analyses, you can do covariate analyses to be able to see if there's a statistically significant covariate effect on different PK parameters, and that type of approach would enable you to say that if there is or is not an effect that you then would want to account for in your simulations moving forward, that would then take out any specific bias and account for that in the model, so that you're not necessarily seeing that at the end if you stratify by gender.
The other point to make is, we showed you the final model in terms of that anchor for the pediatric DVT patients, but there was a lot of analysis considering other covariate effects, and what you saw in terms of the initial simulation exercises, we used the final model so that there wasn't any place where we assumed that other previously unidentified covariate would have impacted the bias. And for other parameters such as the endogenous anti-10 activity, that specifically was a place where we looked at, is there any other demographic consideration that would impact that? But that was all, studied during the exploratory model building phase, and we didn't see anything relevant.
Thank you, Jeff and Kyla. I have no more questions from the participants. I'm just going to say thank you very much for taking off the time. and effort to prepare this webinar for us. I want to advocate that if any of the participants still have questions, please send me an email and I'll pass them on to Kyla and Jeff. And also I want to add that this webinar recording will be made available on our website, www.pmxafrica.org. And for those of you who have attended, I will pass on the code that Jeff and Kyla have alluded to in this presentation. So thank you very much for joining us.